Let's get warmed up for Biochem 14. First question, describe what light reflexes will be seen in both eyes if the right optic nerve is damaged prior to the pretectal nucleus. So this is also known as an afferent pupillary defect. So when you shine light into the right eye, there's no constriction of either the left or right pupil. But when you shine light into the left eye, both pupils constrict. Next, what are the major ECG changes seen with a myocardial infarction? So you can see ST segment elevation of at least 1 millimeter in 2 contiguous leads. You can see T-wave inversion. You can have a new left bundle branch block. You can have new Q waves, so Q waves that are at least 1 block wide or a third of the height of the total QRS complex. But remember, you only see these changes about 50% of the time when a patient's having an MI. So just because the ECG is normal doesn't mean you can rule out an MI. And the last one, what are the clinical features of neuroleptic malignant syndrome? So you're going to see delirium and mental status changes early on. You can see autonomic instability like tachycardia. Then you're going to see muscle rigidity, myoglobinuria, and hyperpyrexia. Okay, let's move on to the lecture.
Hello everyone and welcome to our Step 1 video on amino acids and nitrogen. You won't need a bunch of tryptophan to sleep through this one. Ha 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 ha. I kid. I mean, amino acids are fascinating. What with all the pathways and enzymes and cycle thingies. Woohoo! Okay, really. Go get some coffee, an energy drink, or maybe that totally legit methamphetamine that your doctor gave you for your intermittent attention problem. Yeah, I know. All right, so now let's talk about protein and amino acid metabolism. Now, as always, biochem is a bit rough. You may need to spend a little extra time after the video to memorize a lot of the enzymes that we're going to talk about today. So first we have our amino acids. Let's talk about these. What are the essential amino acids? What does it mean to be essential? Are they more important? Well, the answer is not really. It's a little bit of a misnomer. But the essential amino acids are the amino acids that need to be acquired through the diet. We can't synthesize these at all. We have to eat them. Now, a great mnemonic for essential amino acids is Private Tim Hall. So the Private Tim Hall mnemonic is PVT, and then the TIM for Tim, and then Hall is all the letters except for the A. So PVT for private is going to be the phenylalanine. The V in private is going to be the valine, and then T is for threonine. And then we move on to TIM, TIM. That goes for tryptophan, isoleucine, and methionine. And then Hall is for histidine, leucine, and lysine. So a good way to remember all that, use that Private Tim Hall mnemonic. So this now brings us to the concept of acidic versus basic amino acids. So let's talk about your basic ones first. So lysine and arginine are particularly basic. They have an extra ammonia group, and histidine is also a basic amino acid as well. Arginine and histidine are particularly important during periods of growth. And then arginine and lysine are important for nuclear localization signals. Arginine and lysine, again, are found in high concentrations in histones. Why is that important? Well, because of their positive basic charge, they can interact with the negatively charged DNA. And then your acidic amino acids are aspartate and glutamate. And that's actually a little bit easier for me to remember because they're also referred to as aspartic acid and glutamic acid. So they're negatively charged at the body pH, whereas the basic amino acids tend to be positively charged. Now, one exception to this rule is that histidine, although it's considered a basic amino acid, has no charge at body pH. All right, so now let's get into some harder stuff. Derivatives of amino acids. So let's talk about those derivatives. What are some of the interesting compounds or things that your body can make with amino acids? Now again, this is a pretty high, probably a 5-star topic. You're gonna have a question about amino acid derivatives. So first let's start with phenylalanine. Phenylalanine plays an important role in the catecholamine synthesis pathway. The amino acid phenylalanine is essential for the generation of tyrosine. Tyrosine is found in high concentrations in thyroglobulin, and then you iodinate those tyrosine residues to make thyroxine, so your thyroid hormone, so very, very important. Now, tyrosine can also be converted to DOPA, and DOPA can be converted to melanin. Now, where in your brain might you have high levels of melanin because of high concentrations of DOPA and dopamine? That would be your substantia nigra, which is damaged in patients with Parkinson's disease. They lose the dopaminergic cells, so they don't make dopamine and they don't make melanin in the substantia nigra. Now, another disease related to this is albinism. If patients can't convert DOPA to melanin, then that results in albinism. So let's move further down here. DOPA is converted to dopamine. Dopamine to norepinephrine is by something called dopamine beta hydroxylase, and that's with the aid of vitamin C. This enzyme is also called dopamine beta monooxygenase, so don't get confused by that. You'll probably want to add that enzyme to your progression in your study guide there. Now, norepinephrine to epinephrine is going to be done with the help of cortisol and something called SAM. We'll talk about that here in just a second. So let's talk about a little bit more about these enzymes and how they're important. Well, remember, phenylalanine to tyrosine is accomplished by phenylalanine hydroxylase. And if you're deficient in that enzyme, you get PKU. And we'll talk about that in the next video. And a lot of these will get a little bit more play in the next video when we talk about the disease states. Not only is phenylalanine hydroxylase needed, but the tetrahydrobiopterin is also required when converting phenylalanine to tyrosine. If either are deficient, then you're going to get PKU. Now, tyrosine goes to DOPA by tyrosine hydroxylase. And then the third enzyme of this pathway that I want you to know is DOPA decarboxylase. And that converts DOPA to dopamine. And that particular enzyme, DOPA decarboxylase, is inhibited by carbidopa, a medication we use to treat Parkinson's disease. Now, dopamine is going to be converted to norepinephrine by dopamine beta hydroxylase with that aid of vitamin C we talked about before. Now, there's something called dopamine beta hydroxylase deficiency where the patient has excess dopamine but a deficiency of norepinephrine and epinephrine. Patients will present with nasal congestion, ptosis, and orthostatic hypotension. And then norepinephrine is converted to epinephrine, and cortisol is going to facilitate that conversion. It's not the actual enzyme, but it helps move it along, of that norepinephrine to epinephrine. So it actually kind of makes sense that the stress hormone cortisol would facilitate that generation of a flight or fight hormone, epinephrine. Now, another cofactor here that you should probably know is SAM. We talked about that before, and that stands for S-adenosyl L-methionine. So SAM is another important cofactor there. Now, what happens when you break these things down? Well, dopamine gets broken down by the enzymes monoamine oxidase and catechol O-methyltransferase, and that's going to lead it down a pathway to HVA. Norepinephrine gets broken down to something called VMA, and then epinephrine to something called metanephrine. Important stuff there. All right, so that all started with phenylalanine. What can you make out of the other amino acids? Well, let's start with arginine. Arginine is used to make 3 different important compounds. It can be used to make creatine, which is a high-energy phosphate carrier that replenishes ATP. It can transfer its phosphate group to ATP. Now, when ATP starts to run out, you have this kind of backup energy metabolite of creatine phosphate that's going to rapidly phosphorylate ADP back into ATP. So it's very, very important. Arginine is also important for the generation of urea, which we'll talk about more in just a minute with the urea cycle. And then arginine can also be converted into nitric oxide as well. And nitric oxide is a smooth muscle relaxer, and it's essential in vasodilation. All right, so now let's talk about several amino acid derivatives that are dependent upon vitamin B6 as a cofactor. So many of our reactions use vitamin B6, and you'll notice in a second that without B6, we would have some potential serious problems. So first of all, B6. Tryptophan is converted to niacin with the help of B6, which is particularly important in the body for the generation of NAD and NADP. So these are essential in oxidation and reduction reactions. Now, tryptophan is also essential for the generation of serotonin, which is a precursor to melatonin. And just as an aside, which gland makes melatonin? Remember, it's the pineal gland. Now, histidine is converted to histamine. Again, you need vitamin B6 for that as well. And then B6 is also needed for the conversion of glycine to porphyrin. And porphyrin is converted to heme. Now, the rate-limiting enzyme for heme synthesis is aminolevulinate synthase. And then the amino acid glutamate is essential for the production of GABA, again with the help of B6 again. And GABA is the main inhibitory neurotransmitter of the central nervous system. So with B6 deficiency, you're not going to have as much GABA around, and that can actually result in a lack of inhibition, which may actually lead to seizures. And then also glutamate is important for the generation of glutathione. Glutathione is an important antioxidant. So B6, super important. All right, so we use our amino acids to make all those substances that we just mentioned. And of course, we put amino acids together to make enzymes and receptors and proteins and lots of other complex molecules. But whenever you break down amino acids, there has to be a mechanism to get rid of the nitrogen. So yes, it's time to talk about the urea cycle. So urea is made in the liver. Now, first of all, you need to know what urea looks like. It's a carbon molecule with a double bonded oxygen and then the NH2 on either side of that carbon. So the rate-limiting enzyme for the urea cycle is carbamoyl phosphate synthetase 1. And it's found in the mitochondria. And that might actually sound a bit familiar because carbamoyl phosphate synthetase 2 was the rate limiter of pyrimidine synthesis. So again, just a contrast, with CPS1, it's used in the urea cycle, it's found in the mitochondria, and it gets its nitrogen from ammonium. CPS2 is used in pyrimidine synthesis, and it's found in the cytosol, and it gets its nitrogen from glutamine. So be aware, they're actually pretty different. Now, I don't think you need to get bogged down in the different enzymes and substrates of everything in the urea cycle. I would know CPS1 in the mitochondria as the rate limiter, and I would also know one other enzyme, ornithine transcarbamylase is another very important enzyme in the urea cycle. So let's go actually a little bit deeper into that one. What happens if you're deficient in ornithine transcarbamylase? That is the most common urea cycle disorder. So this is an X-linked recessive disorder. And if you don't have that enzyme, then you can't undergo the urea cycle. You're going to build up nitrogen and ammonia in the body. Now, this is problematic, obviously, and this becomes evident in even just the first few days of life. All this excess carbamoyl phosphate is converted to orotic acid. So you find orotic acid in the blood and in the urine. Your blood urea nitrogen is going to be actually decreased because the urea cycle doesn't work. You can't make urea. So BOM that we'll see on a lot of comprehensive metabolic panels in the rest of your life is actually an indicator that the urea cycle is working. If it's elevated, you're probably making that urea, but you're not excreting it via the kidneys very well. If BON is decreased, it's probably that you're not making urea. So with ornithine transcarbamylase deficiency, you're going to have symptoms of hyperammonemia, so elevated ammonium levels. You can have elevated ammonia levels either from a particular urea cycle enzyme deficiency or because the cell responsible for the urea cycle is damaged, and this is specifically the hepatocyte. So if you have liver disease, that can cause that hyperammonemia as well. So what are the symptoms of hyperammonemia, that elevated ammonia? Patients will have slurring of speech, they'll have somnolence, vomiting, cerebral edema, and even a blurring of the vision as well. Now, we refer to this constellation of symptoms as hepatoencephalopathy. So again, you can see this with liver disease or if you have a specific urea enzyme deficiency. And again, the most common urea enzyme deficiency is the ornithine transcarbamylase deficiency. So how do we treat this? Well, since this is a nitrogen processing problem, then we try not to increase the nitrogen load by eating a low-protein diet. Now, if it's a urea cycle problem, you can use phenylbutyrate, which binds certain amino acids like glycine and glutamine and helps to excrete these amino acids. Benzoate is also on the list of drugs that can be used as well. Biotin can also be helpful because it actually stimulates ornithine transcarbamylase as well. All right, so that gives you the first taste of amino acids. We'll go over a little bit more in our next lecture on some of the more specific pathologies. But now it's time for that end of session quiz. I want you to try to answer those on your own, and then we'll go through them together. All right, first question. What are the essential amino acids? Remember, we use that mnemonic private Tim Hall. So PVT is for private. So that's phenylalanine, valine, and tryptophan. And then Tim is for the T. First T is threonine, isoleucine, methionine, and then we have the Hall, histidine, leucine, and lysine. So those are the essential amino acids. We have to eat those because we can't make them. Next, what amino acid is a precursor for the following molecules? This is the tough stuff. So first we have histamine. Remember, the precursor is histidine. Next we have porphyrin and heme. So remember, the precursor there is glycine. Nitric oxide, the precursor there is going to be arginine. The neurotransmitter GABA, so the precursor there is glutamate. S-adenosyl methionine, the precursor there is just plain methionine. And the precursor for creatine is arginine. Next, compare CPS1 to CPS2. So that's the carbamoyl phosphate 1 and 2. So CPS1 is part of the urea cycle that we just talked about. And that part's going to take place in the mitochondria, and it gets its nitrogen from ammonium. CPS2 is involved in pyrimidine synthesis. It's found in the cytosol, and it's going to get its nitrogen from glutamine. So very different there. All right, guys, that brings us to the end of the video. I hope you learned something. I'll see you next time.